Okay, we are back and we're here to think about bacterial growth. This ends up being a fairly big topic because we have to understand what bacterial growth is, how we think about it, what cells have to think about. We have to think about how we keep track of it and how we can learn about it. So this may be multiple videos. I don't know how many it will take to get through bacterial growth. But you will discover this as I discover this. So here we go. Bacterial growth is not about a single cell getting bigger. It refers to populations getting bigger. So bacterial growth is an increase in the number of cells in a population. So we think of it as similar to what happens in human cells, where they split into two cells. They get bigger, and then they split into two cells. That's what human cells do when they divide. But human cells have to do mitosis. They have to copy their nucleus. And bacterial cells don't have a nucleus. Prokaryotes don't have a nucleus, so they don't have to do the mitosis part of cell division. But most of what they do is the same. So again, bacterial growth is going to be increasing the number of cells. And the word we use for a cell getting bigger often is elongation, because cells are getting longer. And so we can draw a circle to represent a cell cycle that bacteria might use, but really all cells use this when they are dividing, with very few exceptions. They have to make copies of their DNA. They have to make copies of enzymes. And let's think about what those enzymes would be doing. Now would be a good time to pause the video and remind yourself what is an enzyme. And hopefully what you came up with was an enzyme is a protein that catalyzes a chemical reaction. And hopefully you reviewed what a chemical reaction is or what a catalyst is. But it is a protein that makes a certain chemical reaction go faster. And there are lots of chemical reactions cells have to do. So they have to make new lipids so that they can put them in their membrane to make their membrane bigger. And to make those lipids, they have to break down molecules they bring in and change them into lipids. And that requires chemical reactions. That requires those enzymes. They have to move the lipids to the membrane. That requires enzymes. They have to make the pieces of peptidoglycan. That requires chemical reactions. They have to put them together into peptidoglycan and put them into the cell wall. They actually have to cut little holes in the cell wall and stitch in new pieces of peptidoglycan. That requires a whole lot of enzymes. So they have to make all those enzymes and then do all of those things to make the cell bigger. The cell has to be roughly twice as big as a cell normally would be before it can put half the DNA on one side, half on the other, roughly put half the enzymes on one side, half on the other, and then split into two cells. So generally, elongation happens. The cell copies its DNA. It makes a whole lot of enzymes. It becomes bigger. It does what it needs to become a bigger cell. It elongates, and then it splits into two cells. That splitting into two cells is called binary fission. Binary fission. So fission means breaking, and binary means two parts, something like that. So binary fission is breaking into two parts. So keep this in your mind. Bacterial growth is a word we use a lot, or a phrase we use a lot, and it means the population of cells getting bigger. So the number of cells gets bigger. That's bacterial growth. So the cell cycle, so to speak. All right, so when cells divide, they typically take a distinctive amount of time to do it. Each cell under a certain condition is going to take a predictable amount of time to divide, to grow into a bigger cell, form a septum, and split into two cells. Some species do this quickly, some do it slowly, and depending on the environmental conditions, they may do it slower or faster. But the point is, we can measure how long it takes for them to do that. So let's look at an example, and let's look at how we would graph it. So I'm going to give you a famous example of the bacterial species Clostridium perfringens. Clostridium perfringens is a little bit famous because it set a record for doubling time. So it was known to have the fastest doubling time. Ooh, good idea. Hey, real quick, would a fast doubling time be a low number or a big number? Go ahead, pause the video, think about that, and once you're confident, come back. Okay, so again, the doubling time is the time it takes a cell to divide into two cells. So if it could do it quickly, it's a shorter doubling time. So a fast doubling time is a short doubling time. Back in the 1960s, it caused a massive outbreak of food poisoning at the University of Wisconsin. And the University of Wisconsin, very close to where that happened, had some of the top microbiology researchers in the world. And they were very curious about what caused it, and they learned a lot about it, including the fact that this bacterium, which caused the food poisoning, could double in 7 minutes. And that was the fastest of any known organism could double. It still is among, I'm sure, the fastest anyone has ever found. What I want to do is graph growth. I want to graph what would happen if Clostridium perfringens was growing. And so on the, let's see, move this over here. On the y-axis, we'll have the number of cells. So that's technically the population size. And in units of cells. And then down here, we'd have time in units of minutes. So that's what we're going to graph. So let's say that at time 0, we have one cell. So on this axis, we can go 0 up here. Sorry, I'm writing over my own writing. Sometimes you have to do what you have to do, you know what I mean? So at time 0, we'd have one cell of Clostridium perfringens. And then by 7 minutes, that's the doubling time, by 7 minutes, that cell would have become 2 cells, which would be like here. Well, 7 minutes later, those 2 cells would have doubled again. And 7 minutes later would be 14 minutes. So now they'd be up like 4 cells total. 7 minutes after that, 21 minutes, each of those 4 have doubled again to make 8 cells. 7 minutes after that, 28, each of those have doubled to make something like 16 cells. 7 minutes later, 35 minutes, each of those 16 have doubled, and now we have something like 32. And at 42 minutes, which is 7 later, that 32 would be 64, which is off of the screen. And I'd just be guessing it'd be up here somewhere. And I don't think I can put that point where I want to put that point. There. That's about where it would be. So this gives us a distinctive shape. This gives us a distinctive exponential growth curve. And what we notice about the exponential growth curve, wow, I really messed that up. Hey, it's the exponential growth curve. The slope keeps increasing. It gets steeper and steeper and steeper, but also it gets challenging to keep it on the page. And that is literally the problem we're thinking about right now, is if Clostridium perfringens falls into a vat of beef gravy and grows overnight, it's going way more than 42 minutes. It's going hundreds of minutes, and every 7 minutes the population doubles. So it's going to go into the millions and billions and trillions. And how do we graph all of that? That's what we got to think about here. So exponential growth is a challenge. And if we put a typical growth curve on a typical kind of graph like this, we see exponential growth. And again, it's going to go off the chart. In this case, I made up a data set for us to look at. And in this case, instead of going off the top of the chart, here the numbers at the bottom are so squashed together we can't see what they are. So in this graph, it's the same kind of graph with time on this axis and cells in this axis, but here instead of going up by 1 or 2 or 5, we're going up by 200,000. Add another 200,000, we get 400,000. Do it again, 600,000, 800,000, a million, 1,200,000. So this is very similar to this kind of graph. It's just these numbers are much bigger. So, cool. So what do we do with this? Well, this slide you might notice is asking you what's the population size at this data point. Well, the truth is you can't tell because all these numbers are squashed down at the bottom of this kind of graph. So in the next video, we will look at how we can address that and do some of this graphing. Okay, so I'll see you there.